Mhm. Well, dear students, today we will be tell uh, again about protein synthesis, this glimpse about protein synthesis, and previous lecture was ended by translation. And today we will be tell about regulation of translation, different factors which can influence on it, regulation of totally about protein synthesis, reasons and consequences of mutations, and the diseases associated with changing of genetic materials. Now, and about finishing for translation, different variants of regulation possible. It changing of activity of all machinery of protein synthesis, ribosome functions, and factors necessary for initiation, elongation, termination. Rate of initiation, it important from structure and the properties of messenger RNA, number of messenger RNAs, and abundance of ribosomes, plus additional factors which can influence on extra ribosomal factors. Now about elongation, you know that most important for regulative events, it elongation step. And you know that ribosome consists four sites, site for binding of messenger RNA, site for binding of growing polypeptide chain, P site, peptidyl site, A site, amino acyl site, and E site, exit site. And elongation depend on at first elongation factor thermal unstable, so-called for prokaryotes, or for eukaryotes it elongation factor 2, which after activation binding in GTP bound of amino acyl tRNA, it lead to possibility binding of amino acyl tRNA with a site, with selection of best correspondence between codon-anticodon interactions. Later, elongation factor will be dissociated after hydrolysis of GTP. Later, peptidyl transferase activity associated with ribosomal RNA will be provide interaction of peptide bond between new amino acid and previous. And elongation factor G in prokaryotes and correspondent substance factor for eukaryotes will be associated with a site, and it will be lead to translocation of ribosome along messenger RNA on one triplet, and in P site will be appeared tRNA with amino acids growing peptide will be appeared in P site. Previous P site containing presence tRNA will be appeared in exit site and later will be dissociated. Next step, again, elongation factor plus activated elongation factor plus amino acyl tRNA bound with next free A site, and more will be moved before appearance of stop codon. And the function of elongation factor, one of important factor for regulation of rate of synthesis, and phosphorylated and dephosphorylated state important for its activity. It's possible compared with, for example, synthesis of glycogen, again, glycogen synthase phosphorylated and unphosphorylated, dephosphorylated lead to changing of activity. Also, phosphorylation is a very frequent event which will lead to regulation of synthesis. And plus, any additional factors also can influence on velocity of translation. A glimpse about antibiotics. Antibiotics which can influence on translation, different groups. One of it binding with 30S subunit of ribosome. It, for example, aminoglycosides, for example, streptomycin. It prevent correct interaction codon-anticodon codon-anticodon recognizing, and it lead to appearance of errors in sequence of polypeptide chain synthesis and quick breakdown of incorrect synthesized polypeptides. Other variant of associated also pyrimycin. It you can to see structure with the similarities with active aminoacyl adenylate. Also adenine-like structure, ribose and non-usual binding. And it will be competitor for binding with the tRNA, and complex will be interact with A site and also lead to competition with the normal aminoacyl tRNA. Other group of antibiotics is tetracyclines. Interact with A site also on 30S ribosome, but it will be lead to competition between next aminoacyl tRNA and its structure. Tetracycline and now multiple strains of microorganism, bacteria, adaptate to it, and a lot of derivatives now successfully can to participate in antibiotic activity. So-called microlids. Microlids, you can to see, it consists a long, very big cycle, now with 14 carbons cycle with side groups, and interact with 50S subunit of ribosome, prevent transpeptidase activity, which is associated with 23S ribosomal RNA, and it lead to stop of formation of peptide bonds. Chloramphenicol, also one of ancient antibiotic, interact with binding with 23S RNA with transpeptidase activity. Mechanism action of this other different, but result will be the same. Stop of progression of growing peptide from chloramphenicol and it derivates modern antibiotics. Fusidic acid, competitor with elongation factor G, which provide translocation, activate translocation of ribosome, and binding with its antibiotic lead to stop of translation. Any bacterial toxins, protein toxins, for example, very effective toxin, it enzyme diphtheria toxin with modification of important amino acid radical, modified radical in elongation factor 2, which will be provide interaction next amino acid tRNA. You can to see it modified. Normally, it modified histidine imidazole group, modified imidazole group, all of it called diphthemide. But ADP remodulation of it imidazole group lead to inactivation of elongation factor, and it was very strong, very effective toxin, which lead to blocking and substrate for its enzyme activity, it NAD. You can to see it NAD without nicotinamide. Cycloheximide, again, a competitor for peptidyl transferase. Multiple antibiotics. It reflects a strong struggle against against the bacteria in microbiomes, microbiomes. Next problem, which will be appeared after synthesis of polypeptide chain, is sorting. Each protein will be worked in correspondent place with correspondent environment, and it depends on special signals which will be present in this primary structure. It will be possible to compare it with passenger hubs in airports, for example. It depends on possibility of interaction with and modification with membranes of endoplasmatic reticulum. Any ribosomes can bound with endoplasmatic reticulum membranes, any not, but no, totally inner space of cell can be divided on two volumes, two spaces. It cytoplasm and endoplasmatic reticulum. Nucleus, by the way, is part of cytoplasm because nucleus not separate from cytoplasm by membranes. Membranes with inner hydrophobic space, it a strong factor which prevent free movement of molecules in cells. But nucleus hasn't a membrane barrier between nucleus space and cytoplasm. And it, of course, it will be porous structures with very effective regulation of transport, but not necessary pass across membranes. And well, and you can to see ribosomes very often interact with membrane of endoplasmatic reticulum and inner space of endoplasmatic reticulum, and enzymes associated with membranes of endoplasmatic reticulum. It's important factor for next post-synthetic modifications of proteins, newborn proteins. For example, messenger RNA can be associated with polyribosomal create polyribosomal structure, and ribosomes can be associated with membranes. How it possible? It depends on so-called signal sequence, signal peptide, which will be localized on amino end of newborn polypeptide chain. You know that all all polypeptide chains beginning of synthesis from methionine. Methionine or formylmethionine in prokaryotes, but never never methionine or formylmethionine present on amino end in real working proteins. Why? Because it part amino end of polypeptide chain will be digested, but it part consists information about next localization and working of its protein. When polypeptide chain will be begin of its synthesis, it will be amino end and signal peptide will be appeared in extra ribosomal space at first. Translation not finished, but first signal peptide will be appeared in extra ribosomal space. And it will be recognized by special signal recognized particle. It's relatively complex. You can see there complex structure which consists multiple subunits of proteins and special RNA, which complex specifically bound with 50s subunit of ribosome or 60s, yes, sorry, in eukaryotic events, but take place also in prokaryotes also, but binding not with endoplasmatic reticulum, but with cytoplasmic membrane, secreted proteins. And it complex which recognize signal peptide, will be provide interactions with endoplasmatic reticulum, membrane, so-called translocon, and after it complex with facilitation of interaction with membrane will be dissociated and can participate in next recognizing and binding of next signal peptides. Signal peptides for each protein individual, but it reach by hydrophobic residues and positively charged amino acid residues. And later, polypeptide chain will be synthesized in cistern of endoplasmatic reticulum. And enzymes of membranes of endoplasmatic reticulum, it reached by different membrane enzymes. And enzymes in the space will be provide next modifications. And important in this case, correct folding of polypeptide chain. If proteins cannot correctly folded, it leads to increasing fluctuation of polypeptide chain structure, 2- and 3-dimensional structure. You know that each polypeptide chain try to fold it in structure with minimal free energy, and if it structure cannot be achieved, in this case, this protein will be quickly digested, broken by proteasomal or lysosomal proteases. And enzymes, proteins, sorry, not enzymes, but proteins, which can to provide facilitation of correct folding called chaperones. It different names of it: chaperones, Hedgehog proteins, or sometimes it acute phase proteins. Why? Because chaperone it storage of correct structure and provide folding of correct structure. By the way, if it impossible, in this case, it will be ubiquitinated and broken by proteasomal degradation, or if it in endoplasmatic reticulum, it will be broken by lysosomal degradation. But chaperones, it cylindric-like structure with polypeptide chain input in this structure and unfolded, and it will be provide most effective interaction of short distance and long distance interaction in different part of polypeptide chain, and it will be lead to folding in correct configuration. Different types of chaperones may be present. Total name one of it, hedgehog proteins. Why? Because its synthesis sharply increase in appearance of cells in stress situation. For example, increasing of temperature. Optimal temperature for cells, for example, 37 degrees, but if it will be increased up to 40 degrees, it will be non-pleasure and will be disrupt any effective interaction in chemical reactions, by the way. And in this case, it will be necessary change of a set of expression genes, activate adaptation cell to non-usual state, and activation synthesis of specific proteins. Most increasing of synthesis of different proteins lead to increasing possibility of incorrect folding. In this case, hedgehog proteins will be facilitating of correct folding. And it depends on molecular weight. It's HSP 70 family, HSP 60, HSP 90 family. All of it hedgehog proteins. Its synthesis activate in non-usual stress factor. In humans and invertebrates, it called proteins of acute fast because it synthesis increase in inflammation. Again, inflammation it state when necessary to increase synthesis of defense proteins when will be in aggressive environment, and in this case necessary to destroy it factor which will be lead to inflammation, and it will be lead to activation synthesis of corresponding proteins, and level of with proteins it indicator of level of with proteins it indicator of inflammation in the body. In non-specific indicator, for example, one of it classic example it C-reactionable protein CRP, and also a lot of group so-called pentaxins, and it lead to changing of any specific indicators of inflammation. For example, rate of erythrocyte sedimentation. Increasing of it is not specific indicator of inflammation, but it will be indicator about intensity of inflammation in the body. And for example, albumin globulin coefficient relation between concentration of albumins and globulins in blood serum. Also, it correlate with rate of erythrocyte sedimentation and also non-specific indicator. All of it influence on physicochemical properties of blood serum. And polypeptide chain will be transformed post-translationally. How it will be? No, you know that we talked about signal peptide, which will be upstream of working polypeptide chain structure and must be digested. Also, it can be, for example, the transformation, classic example for students, it transformation pepsinogen to pepsin, pro-enzymes to enzymes, or trypsinogen to trypsin. You see it so-called limited proteolysis. It can be provided in endoplasmatic reticulum, cleavage of signal sequence, and it can be take place in different organelles or maybe in the place of action. Next, disulfide bond formation. Sometimes the correct disulfide bond formation depend on more big structure, precursor structure of polypeptide chain with before proteolytic cleavage. Typical example for it you can see on insulin. Insulin chain, insulin protein, it consists of 51 amino acid and consists two strands, two chains, are A and B. A consists 21 amino acid, B chain consists 30 amino acids, and between them two disulfide bonds and one intra-chain disulfide bond. Correct folding depends on presence of additional peptide pro-insulin synthesizer single polypeptide chain, and in this situation possible correct folding of pro-insulin in three-dimensional space and correct interaction of cysteines with forming of disulfide bonds. Later it 34 amino acids peptide C must be digested and appearance of active insulin. Without it, if it will be artificially synthesized A and B chains, possibility of correct folding of a disulfide bonds will be not more than 20–25% from real when will be folded from pro-insulin. It classic example, limited proteolysis and folding and creation of disulfide bonds. Next modifications possible. Modification of amino acid radicals. It may be glycosylation or interaction with any other monosaccharides. Acetylation. No, glycosylation, it takes place for big part of blood serum proteins. All of it, no, practically near all of it, glycoproteins. And glycosylation important for providing of lifetime of its proteins. Acetylation, it may be, histones. Deacetylation of histones lead to more or less compact packing of nucleosomal non-nucleosomal structures. Phosphorylation. You know that dephosphorylation of glycogen, synthase glycogen phosphorylase, and elongation factors for translation. It's effective method for its regulation. And specific modification of amino acid radicals, for example, classic again, it's hydroxylation of prolines in collagen, carboxylation of glutamic acid in factors of blood clotting. And it lost a lot of different variants of modifications. So for example, about proline hydroxylation in collagen. Collagen, it consists molecules of tropocollagens. Tropocollagen, one polypeptide chain, consists near 1,000 amino acid radicals, and it some hundreds time repeating motif glycine, proline in primary structure of polypeptide. And glycine, proline is 300 time repeating. Sometimes instead of glycine or one of prolines will be alanine, can be leucine, lysine, maybe any other, not aromatic. And you know that presence of proline in polypeptide chain lead to bending of polypeptide chain and destroyed, for example, alpha helix and beta sheets in secondary structure. But if proline will be more than half frequent appeared, and its bending will be repeated present, in this case, in three-dimensional space, each polypeptide chain will be folded in winded in fibrillar structure with periodical winding. And the three polypeptide chains will be associated one to other. In tropocollagen present three polypeptide chains associated by non-covalent bonds, non-covalent hydrogen bonds. And increasing, you know, that energy of interaction very weak, and increasing temperature will lead to destroying of its interactions. But for effective interactions between its strands, necessary post-synthetic modifications of prolines, oxidation to hydroxyproline. A little information about it. More than 20 types of collagen, but every time it must be proline must be hydroxylated. Near half of all residues of proline must be hydroxylated to hydroxyproline. It will lead to appearing of new polar groups, and number of hydrogen bonds will be much more. And in normal conditions, if normal modifications take place, it will be lead to dissociation possible, but in high temperature, more than 60–70 degrees. Now you know that if provide boiling of connective tissue in high temperature long time, it will be lead to gelatinization, forming of gel by gelatin. Gelatin is denaturated collagen with forming of net with random interactions between tropocollagen molecules. But it will be more than 60-70 degrees. But if hydroxylation will be imperfect, but for hydroxylation necessary specific redox balance, oxide-reductional balance in intracellular space. And for achieving of it balance necessary vitamin C, ascorbic acid. And if will be avitaminosis of ascorbic acid, its transformation will be imperfect. In this case, polypeptide chain will be synthesized, but post-synthetic modification will be absent. And the temperature of dissociation, temperature of melting of its tropocollagen molecule will be near 40-45 degrees. Its temperature compatible with temperature of human body. And in this case, will be take place clinical picture of scar. Physical inefficiency of collagen. Everywhere where will be any physical activity against connective tissue, will be quick and wound of creation, bloodness, petechiae, ecchymosis, and for example, broken of connective tissue collagen sac of tooth and typical clinical picture of scurvy. Now it without effective post-synthetic modification of hydroxyproline to hydroxyproline. Well, next type of next type of regulation of protein synthesis is transcriptional regulation. It effective example of it is operonal type of regulation in prokaryotes. Now, you know, we talked about structural genes. Expression of structural genes depend on presence of promoter. And we told that in prokaryotes, near half of all genes organized as operonal type. What is it? It any structural genes with different primary structure, its expression depend on one promoter. Promoter 1 and the structural genes can be more than 2. And it different primary structure for structural genes, as a name with cistrons. And what about similarities of its function? All its proteins, polypeptide chains, participate in the same metabolic pathway. Now, for example, LAC aperon, it enzymes which participate in metabolism of lactose altogether. His aperon, enzymes which will be participate in synthesis of histidine. Tryptophan aperon, synthesis of tryptophan. And all of it synthesis provided by the same single promoter region and a single RNA polymerase. It will be synthesized long messenger RNA, and each part of it, messenger RNA, will be can bound with ribosome and synthesize corresponding protein. Totally. Operon consists promoter, operator, and structural genes. One detail. What is operator? Operator, it short place between promoter and structural genes. Not too long, up to 50, 60 maximum. Usually 10, 20 nucleotides. And it plays for specific binding of specific protein which call protein repressor. And protein repressor, information about protein repressor present in specific part of its genome, so-called regulatory gene. And appearance can be divided on two groups. And it model we described, proposed, Frank Jacob and Jacques Monod, model of Jacob and Monod. And appearance can be divided on two parts. It inducible appearance, and its genome must be present in genome, but usually off. Basal state switch out, but will be activate when it's necessary. For example, E. coli or prokaryote live in situation when source of energy will be glucose, optimal source of energy. But if will be glucose absent, but appeared new other source of energy, for example, lactose or raffinose or any other, it will be possible to synthesize, and important synthesis of it enzymes for metabolism of, for example, lactose, and activate it operon. And other group, it's repressible operons. Sometimes normal basic state, it switch out, off. But, for example, E. coli live in situation environment with absence of histidine, and it will be synthesized from glucose and, for example, nucleotides. And but for it necessary synthesize correspondent enzymes to have genes and a lot of energy for support of it. But when end product target histidine will be appeared, in this case, possible to switch out of it genes and storage of energy and use it for any other activities. And all of it based on possibility of binding of RNA polymerase. We told that sigma subunit recognize promoter, and promoter consists two important essential parts, so-called Lipnov book, minus 10 nucleotides and minus 35 nucleotides, and additional. All of it important part for recognizing of sigma subunits. And other subunits will be interact with forming of RNA, active for RNA polymerase, and will be beginning transcription, but necessary pass across short path which call operator. And promoters can be strong and can be weak. Strong promoters recognized frequently by RNA polymerase. Weak promoters recognized by rarely. Affinity to RNA polymerase for weak promoters will be weak, will be slow. Well, and now about inducible appearance. Promoter, RNA polymerase can bound with it, but operator will be interact and bound with protein repressor. Repressor, information about repressor present in regulative gene, which will be present in this genome, but not associated, can be not associated with operon, not part of operon. And repressor bound with operator, and translation is absent. You can to see operator is a short part, it near 10 now. For E. coli, it near 10 nucleotides, which will be bound with repressor. Repressor it multi-subunit structure, low molecular weight proteins, which will be associated and forming of active form. Repressor is active. But when in environment appeared lactose will be interact with repressor, and the repressor will be dissociate, no, inactive. Inactive repressor will be lead to inactivation of repressor will lead to opening of operator, and RNA polymerase can pass across its place, and the synthesis of messenger RNA is corresponding synthesis of corresponding enzymes. Now you can see in lacaperon it's 3 polypeptides, it's galactosidase for interaction with galactose. It permeases, which provide permeability of lactose, and acetylase, which prevent in non-pleasure configurations of aldohexosis. But if in environment will be present glucose and lactose can lead to opening of operon, but effective transcription will be absent because promoter of lacaperon in normal conditions is weak, and recognizing and binding of RNA polymerase with it promoter will be not non-active. Now you can see two different genes, and one of it affinity to RNA polymerase high, and number of messenger RNAs more, and products will be more. And if weak promoter, in this case, concentration of products will be low. But in promoter region present specific CAP site, so-called. CAP site is site for binding of catabolite gene activator proteins. Proteins can interact with DNA and transformate weak promoter to strong promoter. But activity of its proteins depend on concentration of cyclic AMP. Cyclic AMP is modified AMP when phosphate group interact with third and fifth carbons of ribose. It's strong intracellular mediator. And now, when cells will be appeared in stress conditions when lack of glucose, lack of main source of energy, decreasing of ATP synthesis, and part of ATP will be transformed to cyclic AMP. And cyclic AMP will be activate catabolite activated proteins. It will be interact with CAP site of promoter, upstream of promoter, and transformate weak promoter to strong promoter. And intensity of synthesis of messenger RNA will be strongly activated. It transcriptional modulation of regulation. It additional factor which will be checked. Is energy really low? First, it state of operator, first factor of regulation, and second, it affinity of promoter depend on CAP site binding with catabolite gene activator proteins. It inducible operons, repressible operons. It histidine, tryptophan, and any other operons which will be provide coding of enzymes for synthesis of any known amino acids. And also mechanism practically the same. It also presents promoter. Now you can see his operon promoter, place of binding RNA polymerase, operator between promoter and coding path, and repressor which can be bound with operator. But now in native state and normal state, repressor inactive. It will be synthesized and gradually broken by proteolysis. But it any concentration of repressor must be present, inactive repressor. But when in environment appeared histidine, histidine will be bound with previously inactive repressor and will be bound with operator, and it will be lead to stop of transcription. That's all. Decreasing of histidine concentration will be lead to opening of operator and synthesis of messenger RNA. Now you can see nine genes present in his operon. And now at the bottom you can see reactions of transformations for synthesis of histidine in E. coli, necessary phosphoribosyl pyrophosphate, ribose 5-phosphate after pyrophosphorylation. It's a result of synthesis of pentosophosphate pathway. Next, binding with ATP and nitrogen of adenine nitrogen base will be, after next transformations, will be transformed to imidazole group and the synthesis of histidine. But for it necessary a lot of big part of DNA, necessary a lot of energy for it, transcription, translation, and that. And in this system also present additional modulated factors. But in contrast with lac-operon, modulation take place in his translation for his operon. We can to see in his operon present 4 possible to tell genes or elements which call L, G, D, C. It's very interesting structure with complementary sequence of D part with C and G, and special L leader part. What about it? It's a system called attenuator. Attenuator is a system for checking is the concentration of histidine real low. It depends on L part of L fragment. L fragment, leader fragment, consists a lot of triplets with coding of the same amino acid, histidine. CAC, CAC, CAU, you can see in genetic code, all of it coding of histidine. And when will be beginning of translation, after opening of concentration of histidine decrease, and decreasing of histidine, and the repressor will be open, active. But it additional factor for checking of concentration of histidine. If histidine enough, low but not too low, in this case, necessary to synthesize polyhistidyl peptide, now 7 times. And if concentration low but not too low, ribosome will be passed across this path relatively quickly and will be achieved fragment G. I told that D sequence complementary C with possibility of forming of loop. It loop indicator of finishing of translation. It's signal for ribosome for dissociation, so-called terminal loop, you can see there. And ribosome will be achieved G fragment, will be terminal loop and dissociate. Next translation will be stopped. But if a concentration of histidine real too low, ribosome will be moved along L fragment very slowly. And in this case, D will be interact with G non-terminal loop. It will be also loop, but not signal for finishing of translation. And the ribosome will be gradually pass across its path and will be achieve structural genes and will be synthesis of structural genes. It checking is concentration of end product target of it. Synthesis is really low. Well, and about expression of gene expression in eukaryotes, we will tell after a short break, please. Some break, some minutes, please. 7 minutes, please. Well, for regulation of gene expression, it's possible by different steps. You can to say transcriptional control, um, control of processing, transformations, excision of introns, and transport of RNA to cytoplasm, and lifetime of messenger RNA. You know that now it's effectively used by using of CRISPR-Cas9 system with microRNA labeling and breakdown of messenger RNA, so-called knockout methods. And our genes can be divided also on two groups: constitutive genes which can be synthesized active all lifetime, so-called housekeeping genes, which provide basic functions of cells. Usually it enzymes and systems for protein synthesis, storage and transfer of genetic information. And for living organisms, for humans it up to 3,500 genes from 22,000 of total genome. But minimal set of genes, now it as was provided by Craig Venter, very effective scientist, which it was first determined full human genome. Minimal set of genes in Cynthia 3.0, for example, in artificial living organism, consists some more than 450 genes. And it constitutive genes and inducible genes, which can be activated when it's necessary, and only different types of cells and tissues. Now, for example, regulation provides a total picture. It in cells present in nucleus corresponding group of genes, but it will be inactive. Activation will be provided by activation of transcriptional factors, which will be phosphorylate or dephosphorylate, and signal will be appeared from any growth factors. No, really protein structures hormones, and it will be recognized by membrane receptors. Receptors will be activated by phosphorylation or dephosphorylation, and after inner chains of membrane-associated proteins will be transferred to intracellular chain of factors, enzymes. One of it, for example, activation of MAPK, it mutagen activator protein kinase, activated by activators of mitosis. It will be transfer of cell to next step of cell cycle. And all of it will be lead to activation of transcription corresponding relatively big groups of genes necessary for specific activity of cell. And we told that regulation of the transcription by RNA polymerase depend on multiple factors which will be bound with RNA far from promoter region. Sometimes it may be localized upstream or sometimes downstream in introns on any thousands and 10,000 nucleotides. It place of DNA sites called enhancers. Enhancers, which will be specifically recognized proteins, will be can activate of RNA polymerase. We told that basal state of transcription for eukaryotes, it switch out off, but it necessary a lot of activator factors which will be facilitate of transcription by RNA polymerase in promoters. any enhancers will be interact with proteins which will be receptors for steroid hormones. Now, for example, mechanism action of steroid hormones. Steroid hormones, fat soluble, hydrophobic, can pass across cell membrane without special carriers. And hormone, after appearance in cytoplasm, will be bound, can appear in nucleus, sometimes can interact with receptors in cytoplasm, but specific receptors will be recognized and bound with steroid hormones. And it complex, after activation, usually it dissociation of repressor subunit, will be interact with one of enhancers, which will be localized far from promoter region. It enhancers called HIE, total hormone responsive elements. It may be steroid responsive elements. Can be also retinal responsive elements, which interact with any other factors. You can to see all of it will lead to activation of transcription and activation of RNA polymerase and synthesis of polypeptide chain. And sometimes products of activation of genes will be activators of transcription of other genes. And sometimes it can be very complex picture, very complex when products of one gene will be activate other expression of other genes, and it will be and alter and vice versa. And it will be lead to it very interesting and not fully understandable picture. It's one of region of so-called molecular medicine now effectively try to study it. For example, transcription factors of chondrogenesis, multiple activation of multiple genes, when products of any genes will be activated of any transcription of other genes. Also, studying it one of factors which will be lead to activation synthesis of any groups of proteins, and it will be lead to forming new synapses, or can be facilitate interaction between different synapses, increasing number of receptors and other. And it, when studying, lead to more effective interneuronal interactions, and it lead to forming so-called long-term memory and experience, and multiple repeating of any information will be lead to more effective reproduction of it by forming a new interneuronal nets and interneuronal facilitation of exchange of mediator and receptor factors. Well, factors which can influence on changing of genetic information. Changing of genetic information, total name it mutations. And different types of mutations take place. It can be gene mutations, changing in sequence in a single gene. It can be genomic mutations, changing of number of chromosomes, for example, polyploidia, increasing number, or aneuploidia, missing of any chromosomes. It can be chromosomal mutations, changing in structure of chromosomes, addition or missing of any part of chromosome, transfer of any part of chromosome on other chromosome. Can be cytoplasmic mutations, when nuclear set of chromosomes without changing, but changing in mitochondrial DNA mutations. Different variants. And if you will tell about gene mutations, changing in structure of single gene, it can be changing, for example, purine bases on purine bases, for example, guanine to guanine, or pyrimidine to pyrimidine. It will be transitions. Can be changing purine to pyrimidine or alternative. It will be transversions. Result will be, it will be changing of amino acid sequence, but if you will be remind so-called properties of genetic code or degenerative code, it not every time lead to changing of sequence of amino acids, but it can lead to errors to replication. Also, can be deletions, missing of any nucleotides or growth of nucleotides, or can be insertions, addition of nucleotides. It can lead to changing of frame shifting of frame of transcription and translation, and it will be lead to changing not random but changing of sequence of triplets, and in this case there may be termination, non-planar termination, appearance of stop codon, or can be changing of primary structure, full changing of primary structure of polypeptide chain. It reasons of which will lead to mutations. No, it natural reasons. We told that appearance of oxygen can lead to spontaneous deamination and changing of amino group to oxygen. In this case, for example, for cytosine, it will be changing to uracil. Uracil is non-usual nucleotide and will be exquisite in reparation systems. But if will be methylated, cytosine will be methylated. We told that methylation one factor for regulation of transcription. Methylated nucleotide signals for interaction with proteins which will be recognized and bound with DNA. In this case, oxidation of methylcytosine will be transformed to thymine. It normal, usual nucleotide, and reparation system can not every time can recognize it normal thymine, but alternative guanine can be exquisite and thymine will be present. It normal usual native reason of mutations. Also, maybe deamination, oxidative deamination of adenine, guanine will be lead to hypoxanthine, xanthine, but like acidic bond between deoxyribose and purine bases relatively slow and can be spontaneous hydrolysate. And in our genome, some thousands of purine bases can be spontaneously hydrolysate depurinization, provide the missing of purine bases. Of course, reparative systems will be reparate it, but it again possible if will be other strand of DNA will be modified. In this case, it will be reason for appearance of mutations. We haven't in our genome two cells with the same sequence of DNA. Evolution takes place every time, and mutations will be accumulated every time, from zygote to somatic mutations. Other factor for modifications is alkylation by, for example, methylation or any other spontaneous transformations. It may be result of tobacco smoking. Ultraviolet, important factor for modification. Light ultraviolet absorption by pyrimidine nitrogen bases can lead to binding of covalent interactions between two pyrimidine nitrogen bases, for example, between thymines or between thymine and cytosine, and it also important factor for appearance of mutations. Of course, we have effective systems for reparation of DNA, double strand of DNA, it effective factor for re-synthesis of complementary chain of DNA, but double strands of DNA, double strands of broken of double strands, most dangerous factor for appearance of mutations. But all of it lead to effective changing of between genes, genetic set of genes and sequence of genes and its activity in different humans lead to genetic diversity. And now take place accumulation of information about different genomes, human genomes, after finishing of project of human genome project. And now it take place human diversity project, when for determination of what genes more important for humans or less important, and the mutations in this can be influence on appearance and stability of population. And if we compare sequence of nucleotides and significance for transcription and translation functional elements of genome in prokaryotes, all of it you can see practically all genome important for storage and transfer of information. For humans and for example, now it plants, a big part of genome senseless, and more or less important part of genome, and a lot of so-called pseudogenes can be appeared, and senseless sequence. But now appeared information that is not senseless, fully senseless. Sometimes may be present information about any specific RNAs which we can regulate lifetime of messenger RNAs, for example, microRNAs. Well, now it's information about human genome, nuclear genome. It's near 1.5% of all genetic information. It's important at coding information. Mutation in this part important to lead to appearance of dysfunction of its genes. And regulative parts also near some persons of it. But very big part function it so-called transposons. It's information about ancient factors which will be appeared there, frequently part of transfer by viruses, and it dynamic state. It constantly appeared and missing of any genes which will be moved along genome and can to change it. But mutations in these regions don't lead to any consequences, clinical consequences. And in mitochondrial genome, you have 3.2 billion nucleotides. But in mitochondrial genome, it's 60,500 nucleotides. More than 90% it sends information. Near 5% it regulative parts, and only near 2% of mitochondrial genome information can be changed, and mutation of it don't lead to any consequences. And by the way, it part used for identification of different parts of different personal identifications and determination of evolution of genome. Coding part of genome can be also more than half of all genes function of it known. It enzymes, it regulative proteins, it working with nucleic acids and other. But a very big part function of each genes not determined yet. Its number gradually decrease, but not near 30-35% of its genes function of it unknown, and it's field for next investigations. Analysis of sequence of different genes lead to possible to check roots in different living species. You can to see any part of big part of a genes have origin and similarities with vertebrates. Any part, very big part, it vertebrates and other non-vertebrates. But eukaryotes. Any part the same origin, the same with eukaryotes, not only animals. Eukaryote and prokaryotes near 20-25%. It similarities eu and prokaryotes. Only prokaryotes origin and similarities with near up to 1%, and near 1% absent of any homology with other animals. And it lead to possible reconstructed tree of life in our planet. And you know, you have different three main supergroups of forms of life. It eukaryotes and prokaryotes divide on bacteria and archaeas. Bacteria live in optimal conditions, effective can to work. But archaea, it's thermal living organisms and thermophilic organisms and halophilic organisms which can to live in very high concentration of salts. And eukaryotes, it's single cellular or different groups of eukaryotes, and in this branch, humans localize nearest of yeasts, for example, and plants. You see minimal changing of genome lead to it, and other groups can be genetically far from humans. Now you can to see scale of differences. Importance of mutations and changing of genes for development of diseases. Any diseases, it result of single mutations. Its genetic contributions very high. For example, cystic fibrosis, for example, Huntington disease mutations in correspondent them. Phenylketonuria, of course, and any other. But any diseases, Alzheimer's disease, heart, acute heart diseases, obesity, it also genetic function is very high. Cancer, osteoporosis, asthma, some less, environment conditions more and more, infectional diseases, environment factors relatively high, but genes also important for it. For example, any infectional diseases and sensitivity to it more frequent for any genetic variants. And sometimes it may be environment factors, for example, gunshot wounds and maybe car accidents, depend on environment, but genetic entrance very low. Now you can see triangular structure when monogenic diseases, when one mutation lead to development of a disease, polygenic when multifactorial diseases, when multiple combination of genes lead to development, more or less development, effective development of disease. And different environmental factors can influence more or less effective development of diseases. And a set of diseases associated with mutations. Typical classic example is sickle cell anemia, when mutation in beta globin gene in sixth position lead to decreasing solubility of hemoglobin after reduced hemoglobin associated in strand-like structures, fiber formation, which lead to deformation of erythrocytes in reduced state, in excited state in arterial blood. It can be normal, but in venous blood it will be deformed and lead to microthrombosis and other non-pleasure consequences. It depends on mutation and changing of glutamate in position 6 in beta chain of beta globins, and changing to valine. Now you see a mutation in triplet CTC, it's coding of glutamic acid, but if will be mutation in template strand, instead of adenine will be thymine, in messenger RNA will be triplet CAC, and CAC coded valine. Valine is hydrophobic amino acid radical, and in this case, instead of glutamate, hydrophilic, in reduced state, it will be changing of configuration and sedimentation of hemoglobin in reduced state. But it non-pleasure mutation, it's pleasure for adaptation to malaria. Malaria plasmodium cannot to develop in these erythrocytes, and for example, homozygotes of normal hemoglobin A will be died from malaria. Homozygotes of sickle cell anemia will be died from thrombosis, from complications of its mutation. But heterozygotes can be achieve reproductive age and have next generation. Next genetic disease associated with hemoglobin is thalassemia. Thalassemia is disturbing of normal forming of hemoglobin A. We have a group of cluster of globin genes, beta globin genes, and it epsilon, gamma, delta, and beta, and adult hemoglobin realize variant beta globin. But in embryonal time development will be synthesized other variants of globin genes in chromosome 11. And cluster of alpha globin genes consists zeta, two variants, zeta and alpha 1, alpha 2. And adult hemoglobin realize alpha 1 and alpha 2 variants. Zeta expressed in embryonal period. And in embryonal development, we have hemoglobins, for example, zeta 2, epsilon 2, Gover 1, Gover 2, alpha 2, epsilon 2, Portland, zeta 2, gamma 2, all of it with increasing affinity to oxygen. In fetal hemoglobin, alpha 2 gamma 2, and adult hemoglobin, main hemoglobin for adults, it's alpha 2 beta 2. Sometimes a little we can synthesize alpha 2, and delta 2 subunits with increasing affinity to oxygen. And normally alpha hemoglobin A more than 90%. It optimal hemoglobin for transport of oxygen in altitude up to 2.5 altitude thousands meters. But in mountains, in altitude more than 3 and thousands meters, more effective with decreasing of partial pressure of oxygen. Necessary to synthesize other variants of hemoglobin, increasing synthesis of alpha 2 hemoglobin and fetal hemoglobins. For example, in alpinists which will be adapted to high altitude must be live some weeks in situation in camps in high altitude, which will be try to achieve, for example, Everest or Elbrus or any other high tops of mountains. And synthesis of, for example, hemoglobin A, it combination of expression alpha globins, 2 alpha globin genes and 1 beta globin genes. And the result will be the same concentration of alpha and beta subunits, and if it will be disturbed, will be increasing more alpha than beta, it will be lead to dysfunction of hemoglobin and forming variants of alpha 3, 1 beta, alpha 4, it will be alpha thalassemia. Thalassemia is decreasing of lifetime of hemoglobins, hemolytic state, and specific shape of erythrocytes, thallus-like structure. Or if we'll be increasing beta in contrast with more than alpha, in this case it will be beta thalassemia. It also lead to some similarities of hemolytic state. Analysis of primary structure of hemoglobins and genes of hemoglobins lead to possibility of reconstructed of evolution of its genes. And sometimes you can to see alpha and beta subunits divide one from other. You can to see more than 400 million years ago, after, for example, Cambrian explosion. And sometimes ancestral globins with, you can to see myoglobins, cytoglobins, and neuroglobins have also total precursor. It's interesting, but not such interesting, but not such fully understand picture. Mutations in genes of enzymes can lead to enzymopathies, when a lack of enzyme activity will be lead to accumulation of precursors and decreasing of products. And it will be lead to some symptoms and symptom complexes. And most frequent enzymopathia is deficiency of glucose 6-phosphate dehydrogenase. It associated with X chromosome, and it lead to decreasing of pathway of pentosaphosphate pathway. Now I can to remind glucose oxidized by at first phosphorylation by glucokinase, and glucose 6-phosphate at point of branching. It go to the, for example, glycolysis, but also can be it's glycolysis. But pathway, pentosaphosphate pathway lead to reducing of NAD phosphate. It used for keeping of oxidoreductional balance in cells, and reducing of NAD phosphate go to the synthesis of fatty acids, cholesterol, and other anabolic processes. And the lack of EAT enzyme, dysfunction of EAT enzyme, lead to decreasing of reductive conditions in cells. And it lead to also hemolytic state for EAT carriers. And again, it's adaptation to regions of hot, humid regions associated with Africa and other with malaria, frequent malaria regions. It's adaptation to, like sickle cell anemia, adaptation to malaria plasmodium case. Next, phenylketonuria. Phenylketonuria is lack of metabolism of phenylalanine hydroxylase. And each newborn try to test it for a level of phenylalanine and its derivatives. Now you can to see it well known, I hope for you. Phenylalanine oxidized by phenylanyl hydroxylase to tyrosine. Tyrosine is convenient essential amino acid. If enough phenylalanine and enzyme is active, it's non-essential. Next transformations is phenylalanine decarboxylation, transamination with forming of hydroxyphenylalanine. Next oxidation and transformation to homogenizate, and transformation to fumarate and acetoacetate. In fumarate, it glucogenic part of phenylalanine and tyrosine, and acetoacetate is ketogenic part. A lack of phenylalanine lead to phenylalanine hydroxylase lead to accumulation of phenylalanine, phenylpyruvate, and it lead to dysfunction of central nervous system. And in this case, necessary to keep treatment by special diet with decreasing of phenylalanine level, and tyrosine will be in this case essential amino acid. Next, hereditary enzymopathy, it block of synthesis of melanins. Melanins is derivative of tyrosine, and a lack of transformation of dihydroxyphenylalanine lead to missing of synthesis of one strand, it synthesis of eomelanins, dark black color, and other part it synthesis of phenylalanins, red color pigments. And melanins, it defense against ultraviolet, and different combination of both synthesis lead to less or more dark skin. And in equatorial zone, necessary to have dark skin for defense against ultraviolet, but in high altitude in our planet, it's better to have light skin. It's necessary for synthesis of vitamin D. And its combination lead to complex picture with skin color. Maple syrup urine diseases. It disease associated with inefficiency of enzymes for alpha keto acids derivatives from hydrophobic branched amino acids. Leucine, isoleucine, valine, after deamination or transamination, will be transformed to alpha keto acids. And next, oxidative decarboxylation lead to derivatives which can be switching to input in to metabolite of citric cycle and using for synthesis of ATP. But a lack of it will be lack of it enzymes, alpha keto acid decarboxylase, will be lead to accumulation of EAT amino acids and EAT alpha keto derivatives. It lead to serious dysfunction of central nervous system. development delay, and maple syrup disease called because odor of urine, in this case, have maple syrup odor. And it lead to so-called branched chain oxoaciduria and serious dysfunction, lethargia, coma, epistatonus, and other dysfunction of central nervous system. Cystic fibrosis, frequently observed, relatively frequently observed in European. It mutation in chlorine channel, which will be provide transport of chlorine ions, and together with chlorine will be transport of water. Mutations in this, by the way, it's membrane protein with columns of channel, and valve will be open or closed its channel. And dysfunction, mutations in this gene lead to problems with transport of fluorine and water. In this case, it will lead to high viscosity of mucus synthesis by glands, mucous glands in skin, in mucous surface. And very often it missing of triplet, so-called near half of all variants. Now it cystic fibrosis transport proteins. Lysosomal storage diseases. Mutations in genes which digest glycolipids associated with cell membranes lead to accumulation of its non-digested oligosaccharides in lysosomes. Volumes of lysosomes and volumes of cells will increase, and it lead to serious also dysfunction of liver and central nervous system, and patients will be died in child period. It set of diseases. You can see Farber's diseases, Niemann-Pick, and other Tay-Sachs diseases. It orphan diseases, but present in population constantly. Diseases associated with the enzymopathies of urea cycle, and also serious dysfunction as a result of accumulation of ammonia and intermediate substrates of urea cycle. It can be ammoniemia, citrullinemia, uremia, not uremia, argininemia, arginosuccinatemia. All of it will be lead to accumulation of residual ammonia, residual nitrogens in blood. One of treatment in this case can be, you know that ammonia interacts with carbon dioxide with synthesis of glycine, and it used for decreasing of ammonia level by treatment by sodium benzoate. Sodium benzoate, benzoic acid, it's non-toxic for substance in big concentrational region. And binding of glycine with benzoate will be lead to synthesis of eupuric acid excreted by urine. It's one of variant of decreasing of ammonia in blood. And oncotransformations. Oncotransformations, cell cycle depend on function of specific kinases, cyclin-dependent kinases, and its activity depend on binding with cyclins. Broken of cyclins will be lead to activation of its kinases and the transfer cell to next step of cell cycle. And it can be provided by regulation by specific signals from cell membrane receptors and mutation of it, and increasing of its number will be lead to activation of proliferation of its cells. And mutation in intracellular chains of signal transfer, and mutations in transcriptional factors will be lead to activation of group of genes, which will be lead to proliferation. One reasons it mutations in oncogenes. All of it will be lead to oncogenetic transformations. It may be single mutations in coding path. It can be amplification of genes or rearrangement of chromosomes by appearance of viral promoter associated with coding part of oncogenes and non-planet activation of oncogenes. But its activation lead to activation of anti-oncogenes, and oncotransformations possible minimum of two mutations, one of mutation in oncogene and mutation in anti-oncogene, when normal activation of anti-oncogenes will be impossible. Now, guard of genome, it protein of p53, it any non-planet transformations, damage of DNA or any other stress factors will be lead to activation of it, and it will be lead to stop of cell cycle. But mutation in gene of p53 will be lead to lack of its activation and blocking of anti-oncogenic activity. And double mutation in onco and anti-oncogene genes will lead to development of non-planet transformations. But not all mutations lead to diseases. For example, all of us have differences in genome, and it lead to polymorphism. And its polymorphism lead to multiple genetic variants, and it guarantee of living and guarantee of existence of human population. It so-called polymorphism of genes. Now I will be finished. Thank you very much. Goodbye. Тогда на 5. Спасибо. На 9,50 и на